Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I hope you all are fine. So today, for the course of medicinal chemistry, we will discuss about the topic of drug discovery, and this topic will be continuation of the previous lecture on drug discovery, right? So that's the second part of the drug discovery lecture, and the key concepts which we will discuss in this lecture will be the methods by which we will discover the lead, and the second are the stages of drug discovery that are involved, right? So before going to the methods which are involved in the discovery of a lead compound, just for revision, just go back and see. So what do we mean by the term drug, right? So drug is anything that can affect our body, right? Whether it affects in a positive way or it affects in a negative way. Second, we thought the term we used was the hit. So what's a hit? That once you have a large number of compounds, and among these large number of compounds, the compound that shows the desired activity, right? And one thing other which is also important for you to know, that these compounds which you are searching for, this should be new, right? So if you have large number of new compounds, and among these new compounds, one or many of the compounds that show the desired activity, right? So that will be called the hit, right? And among those hit compound, right, so you improve the structure, you change the structure of these hit compounds until you get a compound with maximum positive activity, or with maximum activity with least side effects. Then this compound will be called the lead compound, right? So this is important thing you should know. So far we have discussed about the drug, so what's a drug, and then you studied about the hit, and you studied about the lead compound, right? And in this connection we also saw about how we can use the computational chemistry and how we can make use of the high throughput screening, right? So just we will consider what are the methods by which we can discover a lead compound, right? So the identification of lead and hit compound is very crucial in the drug discovery, because it's the first part of the drug discovery, right? So there are several approaches for lead identification. So you have to identify a lead, you have to identify a hit compound from a large number of synthesized compounds, right? And all of these synthesized compounds have to be screened for biological activity, right? So you have tens of thousands of compounds or more than these compounds. So in order to find a hit and to improve that hit to form a lead compound, right, so you have to screen all of these lead compounds for the biological activity, and before that you have to synthesize all of these compounds as well, right? So it's a very lengthy process to synthesize a large number of compounds, and secondly to characterize all of these large number of compounds and to screen all of these compounds for biological activity, right? So once you get a lead, you can then modify it further to increase its potency. So this process for screening a large number of compounds for biological activity, for that purpose we make use of a technique which is known as high throughput screening, right? So this technique was discussed in the previous lecture, but just for revision we are here describing here that high throughput screening is a method by which we can identify new chemical entities for therapeutic efficacy, right? And the important thing about that, we analyze these compounds which are in large number, in huge number. Approximately you can analyze about 10,000 of compound at a time, right? So this is the efficiency of this technique of high throughput screening, that you can analyze a large number of compounds at a time, right? So once you get a hit, once you identify a hit, it is refined again, and after refining then you check the property again until you get the refined structure with best possible activity, and you get the refined hit which is called the lead, right? The purpose of refining a hit is to get the lead which has improved selectivity. So what do we mean by selectivity? Selectivity for the target with which it will react or with which it will bind, right? So the lead will have the advantage of improved selectivity, improved binding properties, improved potency, improved physicochemical properties. And this process by which we refine the hit to get a lead, this is called hit to lead development. Right, the first step when you have to carry out the lead discovery, first step you have to synthesize the compound. Second step, you have to characterize and screen those compounds. And once you screen, you get a hit. Once you get a hit, you refine it further so that you get a lead with maximum desired properties. Right, so this method of improving hit to get the lead, this is called hit to lead development. So in this method, the HTS or high throughput screening method, this method is very effective and this method is very useful. However, in case if you are searching that they alternate to this HTS, so there are other screening techniques as well which are used for identification of lead compounds. So these include the focused screen, fragment screen, structure-aided drug design, physiological evaluation, and the use of NMR or nuclear magnetic resonance in screening. Right, so all of these techniques can be used, but this technique is most advanced and this is used to the most extent. Right, so another thing that's very important in drug discovery, this is the rule which is known as the Lipinski's rule of 5. Right, so this is also called Lipinski's rule of 5. It's also known as Pfizer's rule of 5 or simply the rule of 5, RO5. Right, so this rule describes, or this rule helps to predict that a molecule, if it has a low molecular weight, then will it probably act as a drug or not. Right, so if you have a structure, if you have a molecule, and by means of this rule you can see and you can predict if this molecule will act as a drug or not. And this rule is very specifically used for the drugs that have to be taken orally, right, those drugs which have to be taken through mouth. Right, so this rule is the rule of thumb to evaluate whether or not a certain chemical compound with certain pharmacological biological activity has chemical properties and physical properties that will make it likely to be orally active. Orally active means that it can be taken as drug through the mouth. So according to this rule, the molecule must have a molecular weight of 500 or less. Right, so if the molecular weight is greater than 500, that cannot be used orally as oral drug. Second factor is that the C log P value should be less than 5. Right, so what does that C log P value? C log P value is a value that is used to estimate the lipophilic nature of a drug. Right, so this lipophilic nature of a drug, this alters the absorption pattern. So we'll discuss it later. Just firstly go through that rule. Right, so this C log P value should be less than 5. Then it should have fewer than 5 hydrogen bond donors, and it should have fewer than 10 hydrogen bond acceptors, and should not have more than 3 rotatable bonds. So here rotatable bonds mean bond with free rotation. Right, see, this is the rule. Okay, I state again that for a drug to be orally active in humans, it must have a molecular weight of 500 or less. Its C log P value should be less than 5. Then it should have fewer than 5 hydrogen bond donors, fewer than 10 hydrogen bond acceptors, and not more than 3 rotatable bond. It means our drug should have molecular weight not greater than 500, and it should not be non-polar. Right, so if the C log P value is up to 5, so if the C log P value is less than 5, it means that it is polar. Right, so if the value is greater than 5, that goes towards non-polarity. So if we say that C log P value is greater than 5, it means our drug is non-polar. Right, it means it is lipophilic. So greater the value of C log P, higher the lipophilicity, or higher is the non-polar nature, and lesser the value of the C log P, the drug becomes more polar. Right, since the oral atmosphere, since the mouth is a polar medium, right? It has saliva, it has a watery medium. So if a thing is non-polar, right, if a thing is lipophilic, then it will not be able to enter into the mouth, because it has to dissolve in the saliva. It has to come in contact with the saliva, right? So it should be water soluble. So for that purpose, we don't want the drug to be lipophilic. That's the reason Lipinski's rule of 5 states that C log P should be less than 5. So lesser the value, more polar the nature of the drug. And if you increase the C log P value, then the lipophilic nature will increase, and which will alter the absorption parameters, means the drug will not be able to absorb in the oral cavity, right? So that's the value, that's the logic why C log P should value be less than 5. So second is that should have fewer than 5 hydrogen bond donors, right? So you want the drug to be absorbed, but we don't want the drug to be permanently bound through the oral cavity. So that's why we have to limit the number of hydrogen bond donors and hydrogen bond acceptors, right? So hydrogen bond donors should not be more than 5, and the hydrogen bond acceptors should also be not more than 10, right? And the molecule should be rigid, right? So by saying that there should be no more than 3 rotatable bonds, means the molecule should not be much more flexible. It means the molecule should be rigid. So if you have a drug with molecular weight of less than 500, and if you have a polar drug, that means C log P value is less than 5, and if it has fewer hydrogen bond donors, it should have hydrogen bond donors or acceptors, but it should not be in greater number, and the drug is rigid, right? So if all of these parameters, all of these 5 parameters are met, then it is said with maximum probability that this drug can be used for oral use, right? That can be taken orally. And the validity of this rule can be seen from the fact that most commercially available medicine have molecular weight of less than 350 and have C log P value of less than 3, right? So the drugs that are available in the market, they have C log P value of less than 5. That's maximum of the drugs that are available in the market have the C log P value, which is the lipophilicity profile, that have a lipophilicity profile of less than 3, means that they are polar, and they are less lipophilic. So now let's go to the method of lead discovery, right? So there are different important techniques that are used in lead discovery. So we will go through these one by one. So first is the random screening. Second is serendipity. Serendipity means by chance, right? If something happens to be by chance, then that's called serendipity. Next, molecular modeling, then molecular manipulation, molecular designing, and the metabolites of drug. So I will see these one by one, right? So firstly, the random screening. This approach is valuable when a known chemical compound of a drug desired activity is unavailable, right? So for example, in the current scenario, so we have corona, and suppose that we don't have any compound which can kill corona, right? So under these conditions, when a chemical compound of the desired activity is not available, right? So this method is some rocket science, not intellectualization. There is no scientific reasoning in this method. So what we do, that all available chemical entities that are available, right? So they all are used to be screened for that activity, right? So this method involves the new chemical entities to be subjected to a wide range of screening tests to establish their different biological activities, right? And this approach is useful for drug lead discovery when nothing is known about the drug receptor, right? So when you don't know about anything about the receptor, right? So the important thing about this method of lead discovery is that, number one, you don't have any chemical compound with the desired property available. Second, you don't know nothing about the receptor target, right? So you don't know about anything about that disease or about that problem, So what is the random screening? Random screening is sort of blind hitting to hit a nail, right? So maybe you keep on hitting at every places, and maybe you get a hit on something, right? So what we do is that whatever we have available, right? So we screen all of those compounds for different biological activities, and we may get a hit for some desired activity as well. For example, let's suppose that we have about 1 lakh different chemical compounds, and we have situation like this coronavirus, right? And we don't have any chemical compound of desired biological activity, and let's suppose that we don't know what are the receptors with which this corona binds, right? So what we do, we take all of these 1 lakh of compounds and we screen them for different biological activities, including the activity that involve this killing or destroying that coronavirus, right? So it is a possibility that among 1 lakh molecules we may get 2 or 3 hits, and it's also a possibility that we don't get anything at all, right? So that's why random screening is sort of blind hitting to hit a nail, right? So you may get a hit, or you may get nothing at all, or you may get a lot number of hits. So examples of medicines that were discovered in this way by use of random screening involved the streptomycin and tetracyclines. So these were the drugs that were discovered by means of random screening. The second method of lead discovery is serendipity, means to discover something by chance, right? So serendipity refers to the accidental discovery that is made while searching for something else, right? Suppose you are looking for something else and you get into something else. Just for example, let's consider that you are searching for an anti-cancer drug, and what you get is something that is effective against inflammation, right? So you don't get anything about the anti-cancer thing, but you get something for inflammation. So this discovery is by chance discovery, right? So serendipity is very important because it has led to introduction of many useful drugs in the past. For example, penicillin. So penicillin was discovered by chance from a fungal fungi which was known as Penicillium chrysogenum, right? So this was a surprise discovery, and that penicillin was found to be an antibacterial agent. For example, other examples other than this penicillin, so we have lignocaine and phenytoin. So both of these are arrhythmics, right? And these were discovered by chance as well, right? So serendipity leads to new drugs being discovered, and it also involves to finding the uses for old drugs. So you can find, for example, an old drug that's available in the market already, so maybe that drug is being used to treat fever, and by serendipity or by chance you may also find out this drug which is treating the fever, this can also be used to treat some other illness, for example, that also used for treating the malaria, right? An example in that you can just relate with the current scenario, that it has been found that the medicines which are used for treating malaria, they are also found effective against this coronavirus. So this finding is serendipity, because this finding is a result of by chance discovery that this malaria can also be used for this coronavirus, right? So serendipity not only involves the discovery of something new, but also involves finding the new uses for all drugs, right? So this is the importance of serendipity. So next method of drug discovery is molecular modeling, right? That's a computational tool for medicinal chemistry, and it is very helpful, especially in the early drug discovery and early development. And nowadays molecular modeling has become the most essential part of the investigation. So this modeling, this computational tool is helpful because it helps in predicting and explaining molecular and biological properties of organic molecules, right? So in case if you don't screen any molecule, just have a structure of a molecule, and what you do the computational studies of that molecule, and by use of these computational tools you just predict whether these compounds, these organic molecules will be used as a certain drug or not. You can predict whether this molecule will have that biological property or not. For example, I have a structure and I do the computational studies, I do the molecular modeling for that compound, and by using molecular modeling I can predict that this certain molecule will be active against bacteria or not. So I have not carried out the actual studies, but by just making use of this computational tool which is known as molecular modeling, then I can just predict, and such predictions are very much accurate as well, right? So this is a very good tool. This is a very good method for the discovery of lead compound. So today it has become trend that even before we synthesize certain molecule, right? So if we have certain molecule in mind, what we do firstly the molecular modeling, and by means of this molecular modeling we predict the molecular properties as well as the biological properties of that molecule. And once we get that, okay, this molecule has that biological activity, then we synthesize that molecule in the laboratory, and then to ensure the study or to authenticate the studies or the result of this molecular modeling, we carry out the biological activity to see for sure if it has that biological activity or not, right? So this method is a very important part of drug discovery nowadays. The next method in the discovery of lead compound is the molecular manipulation, right? So in this method what we do is that we synthesize the analogues of existing drugs and evaluate them for biological activity. Right, so it's more logical approach than the previous ones, right? So before that we saw about how we can do the random screening when we don't know nothing about a disease. Second was the by chance discovery. Third is the computational tool. Now this molecular manipulation, that's a more scientific approach towards the discovery of the lead, right? So what we do, suppose we have some existing drugs, right? They have some chemical structure, so we modify the chemical structure, right? So we say that we synthesize the analogues of the established drugs, right? And when we synthesize the analogues of these drugs, then we evaluate these analogues for their biological activity, right? There is a possibility that among these analogues which we have synthesized, some of the analogues have better effectiveness as compared to the parent drug, right? So it's a possibility that the ones we synthesize have added advantages, such as they might have increased rate of absorption, right? These newly synthesized analogues, it's a possibility that one or more of them have greater potency and greater selectivity and fewer side effects. So most of the synthetic penicillins and cephalosporins have been developed by this method, right? So this is more scientific and logical method than the previous ones. Then we have molecular designing. So this method involves designing compounds to fulfill a specific biological activity, right? That's a rational form of drug designs, and that may involve synthesis of naturally existing substances as precursors for neurotransmitter, right? So in this method what we do, that we must know the receptor, the nature of receptor for a certain disease, right? So if you know about the receptor with which the drug has to bind, right? If you know about the structure of the receptor, what we do, that we design the drug according to that receptor, right? So that's more about the computational studies as well. So what's the difference about this method and the other method is that in this method we know the nature of receptor, and we computationally bind our drug to that receptor, and we just design the drug so that it completely or effectively bind with the receptors, right? So in that way we say that we tailor the drug, we make the drug, right? We design the drug so that it can bind to the receptor, right? So that's most rational form of drug design. The first method was the random method. In the random method we knew nothing about the receptor and we didn't have any available chemical, any available drug for that medicine. Then second case, there's serendipity, that was by chance discovery. Then we move towards something which is computational studies. Then we synthesize the analogues of a medicine which is already existing, and we check that if any of the analogues is more active than the parent drug. So now we are moving towards much more rational designing. So we know about the receptor, and by means of the computational tools we design a structure. Theoretically, we design a structure on the computer in such a way that this drug, this molecule of ours, is able to bind properly with the receptors, right? So this method is known as molecular designing. Last method is the metabolites of a drug, right? So we have already a drug that's used for a certain disease. What we do is that the drug, when we take the drug and it enters into our system, the drug is broken down, or we say the drug is metabolized. And these metabolites of the drug, these broken down fragments of the drug, they are still active, right? It means that these metabolites, they have certain therapeutic effect as well. And sometimes these metabolites, these derivatized drug inside the body, sometimes these metabolites are even more effective than the parent drug, right? So what we do is that we study the drug is metabolized to result in formation of this and this metabolite, right? If we identify the metabolites of a certain drug, then what we do is that we evaluate these metabolites and check if these are better than the parent drug or not, right? The pharmacologically significant metabolite is then subjected for structural modification, right? So once one of these metabolites has been shown to be most effective, what we do is that we carry out the structural modification so that we attain the drug with maximum stability, with maximum efficacy, with maximum selectivity. So examples of the drug that was discovered in this way is nortriptyline. So nortriptyline was actually a metabolite of a drug which is amitriptyline, right? So that was the parent drug, and that drug metabolized to form a large number of metabolites, and one of these metabolites was nortriptyline, and it was found that this nortriptyline is even more effective than this parent drug, right? So these are different 6 methods for the discovery of lead compound. So we can discover a lead compound by random screening, when we don't know nothing about the disease and we don't know nothing about the receptor. The second method is by chance discovery, which is known as serendipity. Then we have the molecular modeling that involves the use of computational tools. Then we have the synthesis of analogs, right, from the parent drug. And then we have molecular designing, which is again a rational form of drug design that involves computational tools. And lastly, we have the metabolites of drugs, and we see that if it's possible that one of the metabolites is more potent, is more effective than the parent drug, right? So after we have known about the methods by which we can discover a lead, let's go towards the process of drug discovery, right? So there are different stages of drug discovery. The first stage for the drug discovery is the identification of molecular target, right? So let's go towards the human body again. So human body functions by means of different biochemical processes which are going on inside the human body, right? And as you know that all of these biochemical processes, they require certain chemicals, right? And most of these chemicals are available inside our body, right? And these chemicals and these reactions, these biochemical processes, they are maintained, they're regulated by means of enzymes and some endogenous factor molecules via their receptors, right? A disease state is identified either by the abnormal biochemical functioning or over or underproduction of some of the intermediates, right? So disease state occurs if a physiological process get balanced, means a biochemical process is not functioning at all, right? Because of any factor or because of the production of some of the intermediates, right? So these biochemical reactions result in formation of certain products, and there are some intermediates of these products. So if in certain cases the intermediates of these biochemical process is not produced at all, or if the intermediate is produced in excess, then in these conditions we have some physiological disorder and we say that disease state has occurred, right? So whenever the disease is occurring, it means there is some imbalance, there is some problem in the biological chemical system, in the biochemical processes that are going on inside our body, right? So firstly, when we have to start the drug discovery process, right, we have to know for sure that what is the site we have to target, where is the problem, right? So where the problem lies, there is our receptor, so we have to target that site where the problem is occurring, right? The problem may be occurring with the enzymes, which are the mediators of these biochemical process, and the problem may be occurring with the receptors, right, which involves the endogenous factors, right, or the problem maybe with the nucleic acids, right? So firstly we have the enzymes, right? So there are three possible targets when we have to identify. The first step is to identify where the problem is, right? So where the problem is, it's our target. So there are three most common biological targets which are focus of drug discovery. The first target is the enzymes, because the enzymes regulate the biochemical processes, right? For example, the inhibition of human dihydrofolate reductase by methotrexate was found useful in retarding the growth of tumors in human, right? So in this case, the target was this one, methotrexate, right? So this was the drug, and the target was human dihydrofolate reductase. So that's the enzyme, right? So the target was that if we inhibit that enzyme, then tumor will vanish, right? The tumor growth will stop. So this was the drug, and this drug targeted this enzyme, right? So in that case, the target is the enzyme, right? The next target is the receptor. So receptor are basically mediator of responses, and receptors are the means by which many hormones and endogenous factors show their responses. For example, if we block the beta adrenoreceptor in the cardiac muscles, it has been found that this blocking reduces the hypertensive state, right? It means that actually this beta adrenoreceptor is responsible for this hypertensive state. So if a person is hypertensive, then what we do is that we block the beta adrenoreceptor. So what we are targeting this receptor, right? The problem is with this receptor. So the target is where the problem lies. Lastly, we have nucleic acids. So nucleic acids are normally not targeted very much frequently, but they are important targets as well, right? So there are three important targets that are considered that we have to identify in the drug discovery process, right? So targeting means to find out where the problem lies. The problem normally lies either with enzymes. So enzymes are not either not functioning properly, or they are not produced in sufficient amount, or they are overly produced, Also, the problem may lie with the receptors. So the receptor may be blocked to cause to cure the disease, or the receptor may be activated to cure the disease. And lastly, we have nucleic acids. Like the first step was to identify the target. The second step of the drug discovery is to confirm whether the target is the right target or not. Right? Because if the target has been mistakenly chosen, that may result in a very disastrous situation. Right? The second step is the validation of biological target. So once the target is identified, it becomes absolutely necessary to confirm that we have selected the right target or not. The use of reliable and suitable animal models and latest techniques in gene targeting and expression are used in this process. Right? And the validation process also helps to find that if the drug binds to a secondary target or not, because as mentioned in the earlier lectures, we mentioned that drugs are not only specific in their action. Right? So sometimes drugs bind to more than one target. Right? So drug act on more than one site. So it's possible, and it also occurs, that drug has secondary targets as well. Right? And because of these secondary targets, binding of drug to that secondary targets, we get some unwanted side or adverse effects, and also we get some secondary effects as well. Right? The secondary or side effects of the drugs are because of those secondary targets with which the drug binds. So the second step of the drug discovery is, number one, to confirm that we have selected the right target or not, and secondly, to identify whether the drug is binding to a secondary target or not. Right? So ideally, the drug should bind to a single target only, but that seldom happens. Right? There's a binding to other targets apart from the current target that leads to unwanted pharmacological actions. So these binding to secondary targets, they cannot be completely avoided, but they can be minimized to a negligible extent. Right? So the major targets, the major receptors which are normally targeted are G protein receptors. So shortly, the G protein coupled receptors are abbreviated as GCPRs. Right? So they're the most common and major targets, right, where the drug binds. So normally, over 30% of the drugs that are available in the market, they normally target these GCPRs or GPCRs, the G protein coupled receptors. Right? So we know that whenever we are studying the interaction of drugs with different targets, so we should look for the GPCR, that most probably the drug will definitely bind with this receptor or not. Right? So when we have to check whether the drug is binding to the receptor or not, or if that's binding to more than one receptor, then we carry out the quantitative polymerase chain reaction. This is known as qPCR. So this is one of the techniques which you use to measure the messenger RNA expression of the receptor. Right? So by this technique, we can check whether the drug is binding with the receptor or not, or whether the drug is binding to more than one receptor or not. So the third step is the lead structure search. So initially we studied about the different methods by which we can identify lead. So here is the lead structure, right? So lead compound is the one that has basic structure requirement for the desired action. Means that's the compound that can show the desired biological activity, right? So lead has the structure spaces for further development. Means the lead compound is a good structure, right? That gives the good results, but it is not perfect. Right? So what we do is that we improve the structure of lead further to give it further enhanced action. Right? So lead compound is always compound with best activity, but best doesn't mean perfect. So what we do is that we have lead compound, and the lead compound has desired biological activity, and we improve that structure further. So when we synthesize different compounds through lead compounds, we make use of HTS, that's high throughput screening technique, and this high throughput screening techniques helps to evaluate the synthesized compounds for the desired biological activity, to identify which of these desired compound derived from the lead is the further lead of our study. right? So we have a lead and we synthesize different derivatives of that lead, right? And we screen all of these synthesized compounds from the parent lead compound until we get a most active compound from all of these, and that will become our new lead, right? So when we are saying about the lead, we have studied that how we can discover the lead. So now we study that what are the different criterions for a lead compound. So criterion for lead compound involves that it should have optimum pharmacodynamic properties, right? So it means that the lead should have maximum efficacy, maximum potency, and maximum selectivity. Selectivity means selectivity for the target. Second, it should have optimum physiochemical properties, mean that should be water soluble, that should be chemical stable. The third important criterion for the lead is that it should have optimum pharmacokinetic properties, means it should be metabolically stable, means should not break down further to form different metabolites, and its toxicological aspects should be studied, means that its side effects should be minimum and its desired effects should be maximum, right? And the chemical optimization potential should be there, means that lead should be such that it can be easily chemically modified, it should be chemically synthesized, and we can form a large number of derivatives for that compound, right? There should be ease in chemical synthesis and should be ease in chemical derivatization. Lastly, the lead should be patentable, means it should be new one, right? So if a lead is already existing, it cannot be a lead. So these are the 5 criterions for a compound to be considered as a lead, right? Should have optimum pharmacodynamic properties, should have optimum physicochemical properties, should have optimum pharmacokinetic properties, and there should be some potential for chemical optimization, and it should be patentable. Right, let's again come back. So the last step we saw in the drug discovery was the search for lead structure. So we have already saw that how we can discover a lead. So we have got a lead, and what we do next is that after we discover the lead, we have to optimize that lead, right? So how we can optimize that lead? Once the lead structure is identified, we have to optimize it. So what here is done, that chemist in close collaboration with different pharmacologists will carry out the structure activity relationship, and after the structure activity relationship, this lead compound will be subjected to synthesis of its derivatives. So we will synthesize the derivatives of this lead structure, right? And we have synthesized a large number of derivatives, and all of these derivatives will be screened for biological activity until we get a compound with best possible desired activity, right? So this is one of the methods which is most scientific methods, and other approaches for lead optimization involves the structure-based drug design, the quantitative structure activity relationship, and computer-aided drug design, right? So along with this method by which we synthesize derivatives and we subject these derivatives for biological activity until we find the best possible activity. So other approaches involve the structure-based drug design, then we have quantitative structure activity relationship, and the computer-aided drug relationship. So all of these stages, such as structure activity relationship, the structure-based drug design, quantitative SAR, computer-aided drug design, all of these you will study further in the forthcoming lectures. So all of these techniques generate a huge amount of data, right? So as this huge amount of data assists the chemist in optimizing the lead to best possible structures, right? So by means of structure activity relationship, by means of quantitative structure activity relationships, and by means of other techniques. So these all of the data that helps us to find the structure with best possible desired action, right? So all of these will be discovered in the later part of the lecture, right? So that's all for the drug discoveries lecture. So in this lecture we have studied that what's a lead, how can we discover a lead. So we have found different methods that are helpful in discovering a lead compound. Then we saw about different stages of drug discovery, right? The first stage for the drug discovery is to find the target. So the target is the place where the problem lies. You have to discover where is the problem. So the problem lies at three different places, right? The problem may be in the form of enzymes. So the enzymes are responsible for biological chemical process, right? So whenever a biochemical process gets disturbed, we get the disease. So this biochemical process can be interrupted or can be caused to be abnormal because of the enzymes which regulate such process. It can be because of the receptors which may be even blocked or activated, or that can lie at the nucleic acids, right? So after we have identified the target, so we have next step is to confirm whether it's really the target or not. And also we have to find out if the drug is binding to only one target or not. So if it's binding to only one target, then that's fine, but if it's binding to other than those targets, we have to identify the secondary targets as well, right? And we have to ensure that there's minimum interaction of the drug with the secondary targets, because more the interaction of the drug with secondary targets, more side effects or more secondary effects will be observed. The third step is to search for the lead compound, right? So we have studied different methods already, that how can we search for the lead compound, and after we have discovered the lead, we optimize the lead. For optimization, we make use of studies such as structure-activity relationship, quantitative structure-activity relationship. Then we make use of structure-based drug design and computer-aided drug designs. All of these are helpful in optimizing the lead to best possible structure, right? So all of these things you will study in future in the forthcoming lectures. So that's all for today. So as far as your assignment is concerned, so you have to answer these following questions, right? So you have to differentiate between the therapeutic effect, the side effect, and toxic effect, right? So you have to give suitable examples when and if necessary. Second, you have to answer that which of the methods of lead discovery is the best method, right? So you have studied 6 different methods for lead discovery that involves the random screening, the serendipity, the molecular modeling, then the other 3. So you have to choose which of the method is the best method near you. So you have to support your answer with suitable examples, right? And you have to give examples of at least 5 different commercially available medicines which were discovered as lead compounds, right? So you have to search that all, right? So you have to answer these questions. And as far as the recommended book is concerned, this is Wilson and Gisholt's textbook of organic, medicinal and pharmaceutical chemistry, and it's written by John M. Beale and John H. Block. 12th edition, Lippincott, Williams and Wilkinson. Thank you.